Thank you all for attending. My name is Leanne Wyvinga-Groot, and I am a member of the US HUPO Virtual Media Outreach Committee. And Jordan Burton is my co-host for today. And as a committee, we thought it would be really fun to bring on the lightning talk winners and honorable mentions from the recent US HUPO conference held in March in Portland, right? So today we have four speakers, all at different stages of their careers, and we hope you enjoy their talk. So I'm going to introduce our first speaker. We'll have about 10 minutes per speaker, and then we will be allowing time for questions immediately after their presentation. And then if you have a burning question that got missed in between the speakers, feel free to still put it into that Q&A, and maybe we'll have a couple minutes at the end, or maybe if you have a question that's for all of the speakers. So put your questions into the Q&A and we'll get started with Dr. Ali Mills. So Ali is a research assistant professor in the University of North Carolina and their proteomics core facility. She received her PhD in Michael Emanuel's lab working on cell cycle regulation and post-translational modifications. Her specialties at the proteomics core include affinity purification, mass spectrometry, as well as PTM and targeted proteomics. So welcome, Ali. Thanks for coming and take it away.
Thank you. Can you see my screen? It's not showing that it's sharing for me.
I can see it on my end.
Okay, great. Thank you so much. As she said, my name's Ali and I work at the proteomics core facility here at UNC Chapel Hill. And I'd like to thank the organizers for the opportunity to tell you about a project I had the opportunity to work on here at UNC with another lab on campus. Again, since I work in the proteomics core facility, the bulk of this work is actually done, my slides won't advance now. This worked earlier. The bulk of this work is actually performed by this talented postdoc, Abid Khan, who's joint in Ian Davis and Brian Strahl's lab here at UNC in the Department of Biochemistry and Biophysics. And he's really done a tremendous amount of work, and I'm really only going to be able to hit the highlights in the time we have today. But he is putting together a fantastic publication that will be out soon, and I encourage you to keep your eyes out for it. So the Strahl lab here at UNC is particularly interested in histone regulation. And as a reminder, DNA in your cells is wrapped around these protein complexes called histones, or made up of histones called nucleosomes. And how tightly wound these nucleosomes are determines how open or closed your chromatin is. And in particular, they're interested in how these histones are regulated to maintain this chromatin state. So histones all have little tails that are highly modified by three different types of proteins. So histone tails can be have modifications added by proteins like writers. Modifications can be removed by erasers, and modifications can be interpreted by reader proteins, which read and interpret the message and recruit proteins and other protein complexes to facilitate downstream processes. Together, all of these proteins help determine the histone code in our cells. And any disruption of this code could distort or completely change the messages in your cell and interrupt normal downstream processes, leading to diseases such as cancer. One example of a histone one example of how these proteins might be mutated in cancer is here, where we have histone H3 and its tail here, and the variety of proteins involved in regulating the modifications on this tail. In particular, we're going to focus on SETD2 right here, which is a histone methyltransferase that modifies lysine 36 on the histone H3 tail. SETD2 is mutated in several different cancers, with most frequent mutations occurring in a type of kidney cancer called clear cell renal cell carcinoma, where it's frequently heterozygous loss of SETD2. And these tumors retain the histone methylation mark that we know SETD2 deposits, but have drastic genome instability defects. So the question becomes, are there other potentially non-catalytic roles for SETD2 in these cells that aren't related to its histone methyltransferase activity? So to answer this question, Abid first asked what SETD2 interacts with, and he utilized a bioID system with an apex tag SETD2 that deposits biotin onto proximal proteins, or proteins that interact with SETD2, and we can enrich for those biotinylated proteins using streptavidin and subject the samples to an affinity purification mass spec workflow. And when we compare samples that express the SETD2 apex construct to a negative control, we see quite a number of proteins that interact with SETD2 in these samples. So we see SETD2, which is expected, as well as known components of the RNA Pol2 complex, which are known interactors for SETD2. But there's also a lot of other proteins here. And to get a better idea of what proteins in this massive amount of data might be of particular interest, we use gene ontology to get an idea of what terms are enriched in these data. We saw several terms we expected, such as DNA replication, chromatin, and transcription regulation, but what was very interesting is that they identified this nuclear envelope term. And when we look back at the data, you can see there's a handful of nuclear envelope proteins here. And for the rest of this talk, I'm really going to focus on the lamins, but they're all part of this nuclear envelope structure. This is particularly interesting because the nuclear envelope, which sits at the periphery of the nucleus, is not only functionally distinct from SETD2, but is also spatially distinct because SETD2 is near actively transcribed DNA, which is thought to be internal to the nucleus. So Abid confirmed these interactions in his IPs via Western blot, but then went to look at co-localization using immunofluorescence. So as expected, the lamins localize around the periphery of the nucleus, as you can see by this almost outline of the nucleus you see in this image here. SETD2, however, has this speckled localization that indicates it's located inside the nucleus. But what was really interesting is when Abid looked at mitotically dividing cells, we suddenly now see a clear co-localization of SETD2 and lamin AC here. And that co-localization disappears by early G1 once the cells have fully divided and entered their next cell cycle. To confirm that these proteins are not just co-localizing, but actually interacting in these cells, Abid used a proximity ligation assay, which results in a single fluorescent point at each point of interaction between your two target proteins. And as expected, we did not see that many interactions in your interphase cells, but there are lots of interactions between lamin B1 in this case and SETD2 in mitotically dividing cells. So in mitosis, the nuclear lamina actually needs to be, or the nuclear envelope actually needs to be broken down so that cells can properly divide and segregate their chromosomes without defects. And this breakdown of the nuclear envelope is mediated by phosphorylation via cyclin-dependent kinase 1. So phosphorylation slowly loosens up this intermediate filament to a point where we have smaller subunits of this structure in the cell so that it can easily divide its chromatin. If you have defects in this regulation, it results in in nuclear defects in daughter cells after mitosis. And interestingly, Abid was able to show that the same was true if you knock down SETD2. So just reducing the levels of SETD2 in cells also resulted in massive nuclear defects. So with that, we used a proteomics approach to assess lamin phosphorylation in SETD2 knockdown. And here you can see that when we assessed lamin AC phosphorylation in SETD2 knockdown compared to control cells, we see a reduction in phosphorylated serine 22. And for lamin B1, we were able to detect phosphorylated serine 23 in control samples, but not at all in the SETD2 knockdown, indicating that this modification is lost when SETD2 is knocked down. So the question then becomes, how does SETD2 influence this modification? And we thought, and Ovid thought that it would be a good idea to see first how SETD2 interacts with these proteins. And he used a variety of constructs to test these things, and I'm only going to have the time to show you data for two of them here today. But using again that BioID system I introduced earlier, Ovid had a wild-type SETD2 apex 2, which is the full-length SETD2, as well as this truncated mutant of SETD2 that is missing the disordered N-terminus, but still maintains the SET and SRI domains. When he does an IP with these constructs, what we found was that the truncated version of SETD2 reduces or loses interactions with these nuclear envelope proteins, lamin A, lamin B1, and emerin notably reduced in the truncated IP. And if you perform the BioID experiment, like we did earlier, and compare proteins that were enriched in the wild type to the truncated SETD2 samples, what we found was that almost all of the nuclear envelope proteins were significantly enriched in the wild type condition compared to the truncated SETD2 condition. And furthermore, so was CDK1, which we know, again, phosphorylates these lamins. So then Ovid tested whether or not these constructs interacted with CDK1 via CDK1 IP. And as you can see here, the wild type CDK1 co-precipitates with, or wild type SETD2 co-precipitates with CDK1, while the truncated SETD2 does not. So I've skipped over a lot of data, but quickly to go over the ultimate findings in the model here is that the canonical role for SETD2 is to, as a histone methyltransferase during interphase to regulate chromatin. And then in mitosis, when the nuclear lamina needs to break down, it functions as a scaffolding protein for CDK1 to facilitate phosphorylation and breakdown of the nuclear envelope proteins. So with that, I'd like to thank everyone again who worked on this project. It was a massive amount of work, as I said, from Davo, and he's done a really great job validating lots of things. And there's so much more data that I was not able to show you yet. So with that, I'll try to answer any of your questions.
Thanks, Ali. That was a great talk. I was wondering what the role of the extracellular matrix is and the stiffness of the extracellular matrix in regulating some of this histone code and the chromatin structure, and does SET2 or SETD2 play a role in that?
That's a great question that I'm actually not sure of the answer of. And I'll definitely have to ask Abid and Brian about that, but I don't unfortunately have that answer.
You mentioned some other proteins beside lamins that are part of the SETD2 interactome. Are there any potential functional roles in chromatin organization and nuclear envelope dynamics that these proteins play a role in as well?
That's also a great question. I would have to dig a little bit deeper. There are a massive amount of proteins that we were able to identify, and this work really primarily focuses on those lamins, but it would definitely be worth seeing whether or not there are other lamin regulating proteins in that data set.
Thank you so much for your great talk, Ali.
Yeah.
I have one question
Next,
maybe I could
go
ask
ahead.
just before we move on, just because I couldn't seem to enter it into the Q&A. So in your volcano plot, the second one where you had the truncated SETD2, you showed that some proteins were actually enriched there. Is there any interesting biology going on there?
Yeah, that's yet again another great question. Biology-wise, I'm not sure, as you know, we're on the proteomics side and I don't get to be involved in a lot of these follow-up experiments, and I'm not sure if they have plans for some of those other experiments, but we were really focused on what was being lost in this situation. So, yeah.
Yeah, I can relate to that working at a proteomics core as well. We can't understand everybody's biology. I totally get that.
Thanks, Allie.
There's, yep.
Next, we'd like to introduce Coleman Nielsen. Coleman is a third-year graduate student in the J.C. Price Lab at Brigham Young University. He studies the effect of ApoE4 on the lipidome and related enzymes. He received his bachelor's in biochemistry from BYU, Idaho in 2022, and is currently keeping his eyes open for postdoc opportunities. Welcome, Coleman.
Thank you. I'm just gonna get my thing started real quick. Okay, can everyone see my slides pretty well?
Your slides aren't up yet.
They're not up yet? Okay, let's see. That's interesting. Oh, 1 second, sorry. I had to share my screen. Okay, there we go. Hi, I'm Coleman Nielsen. I am one of the graduate students at in the JC Price Lab at BYU. And I am going to talk about a little bit about what I study. And so I'll be honest, I actually mostly do lipidomics, but we're interested in using lipidomics in a way to understand the activity of the enzymes involved with the lipidome. And so I like to say that we're using metabolites to study the activity of enzymes. And so it's kind of a way to shoehorn lipidomics into a conversation about proteomics. So the title of my presentation is Enzyme Activity at the Omics Level. Evaluating metabolic pathway differences by substrate turnover and availability. Okay, so first things first, I gotta talk about how I won the competition. So I just noticed everyone was being really funny. You know, I had like a real lightning talk prepared where I was going to talk about my research, and I noticed that everyone was being so funny. You know, saying, you know, everyone said that their name was John Yates, or people were doing rhymes. So, you know, I decided to write a little rap real quick, kind of about what I'm talking about. And yeah, so here's the words to the rap. The big part I want to tell you guys is that this is not going to be super data heavy because we have yet to, you know, right now we're generating our data. But I kind of want to talk about the mathematics behind how our process works. And so I'm going to show you the math today. Okay, so this all starts with mass spectrometry. The reason why I'm starting really simple is because I invited some family members to join this. And so I know they don't know much about that. So mass spec is pretty simple. Basically, you take your analytes, your molecules, and then you put them into the mass spec where they are ionized, and then you can measure the intensity of those analytes versus their mass to charge ratio. Now, unfortunately, or fortunately, you'll find when you take a mass spectrum that you don't get one peak for your molecules. You will get many peaks. Usually the first 3 or 4 are visible in your mass spectrum. And the reason why these other peaks show up is due to the presence of naturally occurring heavy isotopes. So for example, in nature, the most common heavy isotope in biological molecules is going to be carbon-13, which occurs at about 1.07%. And so because this shows up at, you know, because a little bit of your carbon in your molecules are heavy, it turns out that you get this statistical distribution of what we call the isotope envelope. So you have all these different peaks which correspond to the same molecule. It's just that you kind of step down as you get bigger and bigger in mass. We can actually induce this, we can actually induce a change in your isotope envelope for a certain molecule by feeding somebody deuterium oxide. So deuterium is a heavy isotope of hydrogen, and when you drink it, that deuterium actually incorporates into your molecules, and so you can actually shift the isotope envelopes of your molecules in your body by drinking it. So we actually take advantage of that in our lab. We do this for proteomics and for lipidomics. So for me, I'm interested in this because this allows me to see how fast my lipids are turning over in the system. So the way that we do this is we measure over a period of time, as we keep feeding a subject deuterium oxide, we get to measure the transition of their isotope envelopes from light to heavy. And that transition can be used to get the percent replacement rate for those particular molecules. So what that means is it's the molecules replaced per day divided by the total molecule pool, and that times 100, that will give you the percent replacement rate for that particular molecule. And this can happen at the omic scale, which is really, really cool. So here I have two different subjects. We have an experimental case and we have a control case. And you can see that for this particular metabolite, the experimental case has a higher intensity for this particular metabolite. And so with mass spec, it's really, really hard for us to get absolute intensities or absolute, it's hard for us to get absolute abundances of our analytes because the intensity is similar. It's related to the concentration of the analyte in solution, but it's not direct. And so in order for us to get a relative abundance, we need to take two cases and divide the intensity of the experimental by the control intensity. We get a fold change value that we call the relative abundance. So we use that. That's part of the math that I'll show you in a second. I'm going a little bit fast. So, we'll have a lot to talk about. It's good. Okay, so here's the math I was telling you about. So what happens is if we take that percent replacement rate for our experimental case and we divide that by the percent replacement rate of the control case, we get another fold change value. And that is actually equivalent to the molecules replaced per day for the experimental case divided by the molecules replaced per day of the control case. And so that is another fold change value multiplied by the total molecules of the control case divided by the total molecules of the experimental case. And so using some algebra and some rearrangement, we find that the molecules replaced per day for the experimental case divided by the molecules replaced per day of the control case, in other words, the metabolic flux, is equivalent to the percent replacement rate of the experimental case divided by the replacement rate of the control case times by the relative abundance. And so using this simple math, we actually can get the relative metabolic flux for hundreds to thousands of different lipid metabolites in the organism. The reason why I'm focusing specifically on lipids is because lipids generally have a long turnover rate. They're incorporated into your membranes in your body, or many of them are. And because of that, it's hard for enzymes to access them. And so they have a very low turnover rate, similar to proteins. We're talking about days to a month for the turnover of a single for the turnover of the entire pool, similar to proteins, which also have a similar lifespan in the body. So it's actually quite convenient for us to give a subject some deuterium oxide and then try to keep them at that same level of deuterium oxide for the course of a month. And then we can measure by taking samples how that individual, how the molecules in the individual are shifting from light to heavy. And as I said before, that shift allows us to get a view of the relative molecular flux of those particular enzymes. So this is really, really of those particular lipids. And so that's really, really good for if we're interested in measuring the relative activity of different molecular pathways in a system. And so that's kind of how we are intending to use this in the near future. We haven't really figured out exactly how we're going to do this yet. We anticipate this will use linear algebra and statistics, but we are approaching that moment. So you can imagine if we can measure thousands and thousands of lipids, if we are able to use our data to get the activity of specific enzyme pathways, how powerful that could be. If we overlap those pathways, hope is that we can actually measure or derive the activity of either individual enzymes or short individual enzyme pathway segments. And if we could do that hope is that we can use this as a way to, as a very effective way to find potential drug targets. A good example is in the case of, in our lab, we study ApoE, as was mentioned in the beginning. So ApoE is the #1 risk factor for Alzheimer's disease. And so we know that ApoE4, specifically one of the isoforms, is associated with a high amount of neurodegeneration. And so, and also because ApoE is a lipid carrying molecule, we figure that, or we hypothesize that ApoE, part of the reasons why there's degradation involved with it is because of modulations in the enzyme pathways. Hoping that this study that we're doing right now will help us to be able to find enzyme targets for particular, for drugs that we can target with drugs. And so right now, we've been very successful with the finding, because we have to find our lipids first before we can apply this flux analysis to them. Right now, we're able to find between 1,000 and 1,400 different lipids per lipid sample. And so we consider that to be enough for our study. So you can imagine we have all these different lipid metabolites. You can kind of view each of them as their own little location or node in metabolic space, and then you can almost imagine the enzymes as being the connectors between those nodes. And so we hope that by getting flux for all these analytes, we can have a really good metabolic network to help us find it. So we go a little bit further. If we use the idea of kinetic values, so I'm talking about like K constant, K degradation. You guys remember Michaelis-Menten equations. Also, just normal reaction kinetics. We actually can find the K synth and the K deg values for each of these metabolites as well. We can actually plot them against each other. So we can, with a single graph, we can show how specific analytes are regulated differently in different systems using changes. And so that's something we're going to be exploring in our next study as well. Okay, special thanks to the undergrads involved in this project. We have a lot of them. Special thanks as well to my mentor, Rusty Denton, for everything he taught me about lipidomics, and also Dr. Price, and for funding from the NIA. So, thank you very much.
Great. Thanks so much, Coleman. Very good. I like how you introduced the topic very nicely because it's maybe not so familiar to our audience. So that's great. Could you talk a little bit about your study design? I know you said you're in the early stages of collecting your data, but how many are these clinical samples or cell lines? And how many samples do you think you have to collect on to get statistical power?
Yeah, that's a really good question. So we are actually doing this with a mouse model right now. So we have CRISPR mice, which have ApoE, human ApoE knock-ins. And so, yeah, so right now we're working with a sample size of what, 100, let's see. So the entire sample is 128, and so 128 divided by 2, that's going to be how many we have for the experimental block. So we spent a lot of time designing the study, trying to get statistical significance. We're trying to make sure that every time point in our study has at least 2 bioreps, and then most important time points, which are going to be our day 32. I could go into that deeply, and then our day 0 mice, which have no labeling, that's where we get our abundance data from. We have 3 bioreps for those.
Nice.
Yep.
So what kind of tools or equipment are you using for this? Are you doing all the extraction by hand, and then what kind of instrument are you running on?
Okay, that's a great question. So we use the Agilent 6560 series right now for the eye mobility. That's what we're doing with this current study. We're also doing another study, which is similar. It's a regional study, and we're using the 6546 for that because it has a very high acquisition rate. So we love that. So yeah, we do the extractions by hand. We spend about a little bit more than a year trying to figure out the best way to get the most lipids from our extractions, and we figured it out. And that's basically what my undergrads do. They just extract. So,
that's awesome. So those are your robots.
Well, we pay them very well. So.
Oh, that's good. So you mentioned you're using ion mobility. I noticed that you have a lot of lipids coming out at a retention time around 25 minutes. Like you had a lot on your plot there. Is the ion mobility helping you resolve those different points?
Yes, the IM ability helps a lot with that. We found that IM ability increases our coverage by about fivefold. So
Nice.
not
Okay.
fivefold, but 5 times. Yeah, that's fivefold. Yeah, about 5 times. We get about 5 times more data for IM. So it just is really good at resolving things, because we have the extra dimension. So
yeah. Okay. And then one last question from Jordan. He was wondering if there's a specific lipid turnover rate that's the most interesting to you at this point.
So we are interested specifically in cholesterol. That's a big player in our study. We know, well, cholesterol is associated, changes modulation in cholesterol metabolism is associated with neurodegeneration. And so we are really interested in that one. We found some really interesting things about cholesterol turnover. And let's just say, very interesting. There's very stark differences between the different types. And you guys can read our paper to figure out what that is exactly. So.
Okay, on that cliffhanger, we're going to move on. Thanks so much, Coleman. Great job.
Thanks,
And
Coleman.
we look forward to seeing that paper to find out more about cholesterol and Alzheimer's and ApoE4. So I'm going to introduce our third speaker. It's Dr. Weijing Liu. And Weijing is currently the vertical marketing manager with the chromatography and mass spectrometry division at Thermo Fisher Scientific, which is based in San Jose, California. Her focus is native mass spectrometry workflow development for addressing structural biology questions. Dr. Liu holds a PhD degree in physical analytical chemistry from the University of Notre Dame, and she completed her postdoc research in Pacific Northwest National Laboratory. So take it away, Weijing. We're looking forward to this.
You need to unmute yourself.
Thanks for the reminder. Can you see the screen well? And can you hear me well?
Looks good.
Thanks a lot. Thanks for the introduction, and thanks US HUPO for organizing this webinar. Today, I will share our recent progress on the intact glycoprotein analysis using the DIA-PTCR, data-independent acquisition proton transfer charge reduction, for intact molecular weight profiling, some glycoform assignment, and we are also leveraging the native top-down mass spec to extract some structural information. Some common approaches for glycoprotein analysis using mass spec include the released glycans to study the glycan structure linkage, glycan composition. Glycopeptide analysis can get us some glycosylation site information, also some glycan composition information. But due to the diversity of different glycan types, like unlinked glycan, O-linked glycans showing on the right, it makes intact glycoprotein analysis very challenging, especially for the denatured intact glycoprotein analysis. Due to the narrow space between the adjacent charge states, the spectrum usually becomes unresolvable, like a hump. Leveraging the advantage of a native mass spec, the wider space between adjacent charge states, we have some chance to resolve the intact glycoproteins. And in today's talk, I will share our approach for intact molecular weight profiling of glycoprotein, glycoform assignment, and some structural information obtained from the native top-down mass spectrometry. The instrument we are using is the Orbitrap Ascend Structural Biology Edition. It was just released 2 weeks ago at ASMS. There are some features highlighted here benefiting the native mass spec analysis, such as a PTCR reagent ion source. It provides a gas phase ion charge reduction to resolve the complex signals. And the new quad can isolate up to M/Z 8000, which benefits some high M/Z signals from intact glycoproteins. The Orbitrap mass analyzer scanning up to 480K resolution, it provides some isotopically resolved information for intact glycoprotein like below 100 kilodalton. And finally, the ion trap can enable multiple fragmentation techniques, hybrid fragmentation, which really better the sequence coverage as well as provide some structural information. The method we're using here, the DIA-PTCR, data independent acquisition proton transfer charge reduction, is to first, when we see the complex MS1 spectrum, we do a narrow small window isolation using the quadrupole, and we deliver the charge reduction reagent into the gas phase. When the charge reduction occurs, it will display a charge resolved spectrum. And then we move on to the second window. It's just adjacent to the first window, and we do the isolation and gas phase charge reduction again. It will give us another charge envelope. By repeating this process, we are able to deconvolute the charge reduced spectrum from each window, and we composite each deconvoluted spectrum to construct the final molecular weight. Oh, this is a method we call the DIA-PTCR. The benefits of the DIA-PTCR is like to resolve the unresolved spectrum, give more molecular weight information. Here is an example from the human fetching. When we do the DIA-PTCR from 3,300 and 4,500 with 20-something quadrupole isolation, here we show two isolation windows. One is from a relatively abundant MS1 signal, and the right window is from like baseline level signal intensity from MS1 scan. We can get both isolation window well resolved after the proton transfer charge reduction, especially for the low abundant signals from MS1. We are even seeing a second charge envelope. It was hidden in MS1 previously because of the congested spectrum signals. It's not zooming here, but the data was acquired at 480K resolution. It truly enables isotope resolution of each glycoform. And due to the separation of congested signals in the MS2 scan, previously we can only identify 29 glycoforms from the full MS1 scan. Using the DIA-PTCR approach, we can identify over 200 signals like glycoforms or proteoforms from these human fetching samples. Here is another glycoprotein, the spike protein receptor binding domain. Is also heavily glycosylated. It's a small protein, but after conjugated with those glycans, the spectrum from DIA-PTCR becomes really complicated. Oh, it's kind of hard, even difficult to assign the glycoforms from intact level in the control experiment. By trimming off the N-glycosylation using the pangase F, we observe the major loss of the mass shift is around like 4,200 Dalton. By looking at some literature, we know the glycan composition is showing here. And after that, we took the spectrum of the N-glycosylation sample using the 480K resolution and to get the accurate monoisotopic molecular weights. We're able to assign the all glycoforms at intact level. And from the bottom up studies, we notice there are two sequence variants. We are able to unambiguously assign the same glycoforms on the two different proteoforms. When the intact protein are getting larger and larger, all the glyco forms become like more and more complicated. It's not always able to assign the glyco forms on the intact level. Here is a case, it's a sample shared from our collaborator at Genentech in the Fc fusion protein. And on the top region, there are like many glycosylation sites. We received 3 samples with 8, 12, and 15 glycosylation sites. Here showing the unresolved MS1 spectrum. When we do the DLPTCR of this complex samples, we are still able to construct the final molecular weight profile from these 3 samples. And we are able to see some mass shift between like sample 8 and sample 12, indicating that more glycosylation sites actually lead to the mass shift, drifting to the upper end. And by anchoring more glycosylation sites on the biologics, the molecular weight shift is kind of pausing here. It's trying to move upward, but it's like hitting the wall, which reflects some steric hindrance effect there. Oh, in this case, we are still able to see the molecular weight difference reflecting the glycoside effect. However, glycoform assignment could still be challenging for such samples. Switching the gear to some structure study, because we have the multiple fragmentations on this ASCEND instrument. And here is still the human factoring. It has an A chain and B chain linked by the propeptide. The A chain is at the beginning and B chain is at the end of the sequence. However, when we do the native top-down studies, we notice that the B chain is actually linked to the cysteine, very close to the N-terminal on the A chain. It reflects the B chain and A chain is bonded through this disulfide bond to form a loop structure of the factory. Though the native top-down analysis not only confirm the sequence, but can also resolve the chain connection here. Here is another protein, nucleocapsid protein. I ordered it for the glycoprotein analysis, but it turned out this protein is expressed in E. coli. I didn't identify any glycoforms there. However, when I did the ETD activation on it, I got a strong CRN sequence ladder, which means the N-terminals is really exposed of this protein. By reading the literature and looking at the structure online, so it actually correlates with the structure very well because the N-terminus is exposed and the C-terminus is responsible for the dimerization. It's actually like hidden there. Here is a summary. So using the high quad isolation plus the gas phase charge reduction, the DRPTCR approach can streamline the complete molecular weight measurement for glycoproteins. Assignment of some intact glycoforms is doable when the protein is small, relatively small, and the glycoforms are not extremely complicated. But for really large glycoproteins or complex glycans, it is still challenging at this stage. So the native top-down analysis could not only identify the sequence, but also reveal some structure inside of the glycoproteins. With that, I'd like to thank my colleagues from Thermo Fisher Scientific and the collaborator from Genentech.
Thanks, Weijing. I was wondering, in your FC fusion protein example, is there a way to orthogonally separate some of these isoforms for introduction into the mass spectrometry or mass spectrometer to help to kind of separate better the glycoproteins, or is this a problem that's pretty prevalent in native mass spec?
Yeah, I believe the collaborators has some like a SEC fractionation or separated with some ion exchange approach here, because just to reduce the complexity of the whole process, we inject everything into the mass spec and try to study the gas phase fractionation. But that's just a very small portion of this study. The collaborators they have some like data showing the LC fractionation, and then subject to the native mass spec analysis, it could reduce the complexity from there.
I see. And this was you present a new instrument that it sounds like was released at ASMS this year. And I'm wondering, what is the cycle time at 480K resolution for that instrument?
The transient time, I don't have it on top of my mind. It's because on another instrument, 200K is like 1 second or 2 second transient. I think it's like between 2 or 4 seconds.
I see. So
Transient
you're
time.
collecting for quite a while to get that resolution. That makes sense.
For each, for one individual scan, right. It also depends on the injection time and the AGC target.
That makes sense. Leanne, do you have any questions?
Yeah, just wondering, not all of us are lucky enough to have access to this Ascend structural biology version. Can any of these methods, the DIA, PTCR, be adapted onto other thermo instruments, or is it really very specific?
Yeah, we have some like we will have some. This method can be set up on any tribrid instrument. Like if we have Eclipse in our lab, we can also set up this method as long as it's equipped with the PTCR reagent at the front end of the ion source. And the DIA template is always there. We can build up the method from there. So it's not just limited to the Ascend structural biology edition.
Okay, that's great to know because we have a tribrid.
Yeah, that's a great question.
Thanks, Weijing.
Thank you. And I will stop sharing.
Last but not least, we'd like to introduce Noah Earls. Noah is an undergraduate student in the biochemistry program at Brigham Young University. He has worked as a research assistant in the J.C. Price Lab at BYU for the last 3 years, conducting both Alzheimer's and cancer-related proteomics experiments. Noah also serves as the undergraduate representative on the U.S. HUPO membership committee. Welcome, Noah. You're muted.
Yep, there we go. It is an absolute honor to be here. Thank you, everyone. If you caught that, I'm also a member of the JC Price Lab, but don't be afraid because Coleman and I do very, very different projects. So today I'm going to be discussing the tissue-specific degradation of sphingosine kinase. And specifically what we're talking about is this type of drug called a PROTAC. And now a PROTAC is a proteolytic targeting chimera. And what that means is that the PROTAC itself has these specific components. One component binds to an E3 ligase molecule, and there's a linker component, and then there's a warhead. And this warhead can be specifically designed to target a specific area of your specific protein, and it can be very specific and selective. And so it's very optimal to get targeted degradation. Now, the way it works is the PROTAC, when applied, binds to the E3 ligase molecule, and then the warhead allows the target protein to be brought close to the E3 ligase, and the E3 ligase then ubiquitinates the target. The target is then available for the proteasome, and it's marked for the proteasome to then be dissolved and degraded. So the reason our target specifically is sphingosine kinase 1, and it's this weird little suspiciously simple protein. What it actually does, there's a high incidence of this protein across multiple cancers, and it's an established target for cancer therapeutics. There's lots of sphingosine kinase inhibitors out there, but its exact role in cancer is unknown. You can see above in this chart where there's high expression level across multiple cancers, and some of which have much higher than others. Pancreatic cancer is one of those, and we'll be talking a little bit more about that later. So what does sphingosine kinase actually do? Well, what it actually does is it does a simple organic chemistry little phosphate addition. It's just takes sphingosine and it adds a phosphate group to turn it into sphingosine 1-phosphate. Seems, like I said, suspiciously simple, but somehow, and the research is as of yet rather unclear exactly how, it affects cell cycle arrest and apoptosis and cell proliferation and lots of very obviously cancer-related tasks. And that's why sphingosine kinase itself is a established target for developing cancer therapeutics. So we created this task for ourselves. We wanted to degrade sphingosine kinase 1. There are existing inhibitors, but there's not existing degradation mechanisms. So in order to do this, we had to create a PROTAC with a known SPHK1 inhibitor as a warhead, because that inhibitor already binds very tightly to sphingosine kinase. If it didn't, it wouldn't be a great inhibitor. We then want to apply said PROTAC to different cancer types, and then we're going to cure cancer. Hooray, right? A little simplistic, but you can kind of see the goal. So when we designed this PROTAC, we wanted something that could target pancreatic cancer because for those who work with cancer, it's a terrible one. If I'd argue it's the worst, other people might disagree, but I think it's terrible. And it really doesn't have many treatments and its prognosis is horrible. But because it's pancreatic cancer and it's a pain to work with, we also wanted something that could be applied to many other cancer types. So we selected our E3 ligase, which is CRBN. Now, this is present in pancreatic tissue. Notably, if you can see on this graph, on this chart, pancreatic tissue doesn't have a lot of different types of E3 ligases. So you can kind of see as you're designing PROTACs, you can create E3 ligases, you can select E3 ligases that are very tissue dependent. And that is probably the #1 appeal of PROTACs as a drug. But once we created this and started applying it, we ran into a problem. Sphingosine kinase is a low abundant protein. I argue it's a super low abundant protein, and it's incredibly difficult to see through traditional mass spec methods. Now, that's kind of the universal problem of all mass spec kind of experiments is sometimes you can't see what you want to see. So my task and what I presented at HUPO and what my specialty was on this project is to solve said problem. So my first solution is my, little special area of expertise is a data independent acquisition, right? In our lab, we can reliably detect 500 to 1,000 additional proteins every time we run DIA as compared to a regular data dependent acquisition run. We can drastically reduce the effect of replicate filtering. And what I mean by that is when we're quantifying our data, if we see a protein in one sample but in none others, we can't use that protein. And so this fills in those gaps in our data. And then we can improve the resolution of low abundant proteins. That's what I thought we could do. Now, importantly, the reason I thought that is because data dependent acquisition, the traditional method, it works by selecting signal-based. It does signal-based acquisition, and it selects the highest abundant peptides in the MS1 scan to then fragment into MS2. DIA, however, you give it a specific window within the MS1 scan, and it fragments everything, right? You can see lots and lots more proteins. And I expected that we would see lots more low abundant proteins. But when we did this comparison, and on this graph, you can see protein abundance order of magnitude, right? That's the key bottom. So we expected to see in our DIA more E2, E3. We expected sphingosine kinase to be somewhere around that abundance, because like I said, it's a very, very low abundant protein. But we didn't. Fascinatingly, it didn't work. But what we did learn is that DIA is actually more effective at finding middle abundance regions, this E5 to E6 regions. And unfortunately, in our DIA runs, sphingosine kinase is not there. So solution 1, that doesn't work. So we then decided to go to solution 2, which is antibodies. And there's several different experiments. We did Western blots and lots of them. And Western blots actually worked. We were able to see sphingosine kinase and we were able to see concentration changes. But if Coleman explained turnover rate, and that is kind of the Price Lab's modus operandi, right? It's our niche. It's what we do, right? And the key here is that what's the best way to tell if you're degrading sphingosine kinase than measuring if the degradation of sphingosine kinase. And so Western blots won't work for that. We tried immunoprecipitation with specific antibody-bound beads, and that was promising. It was a good idea, but with immunoprecipitation, you're working with such small quantities of protein because the antibodies are expensive. We couldn't realistically make it a feasible method for studying lots and lots and lots of things. And the mass spec couldn't even measure any protein because our amount was too small. We then did in-gel digest. And this method, very similar to the Western blot, it's the same starting process, but instead of running it and putting it on some paper, you actually cut out the area, the band of where your protein of interest is based off of its molecular weight. And then you digest that in the gel and run that on the mass spec. Now the key here is that it actually looked like it worked. Now we didn't really see very good data, but that's an optimization problem. Peaks was able to actually detect sphingosine kinase, and it was able to quantify it, although its quantification was suspiciously bad. So with some optimization, that looks like it might be a feasible method. But had another idea up my sleeve. So there's this method called stable isotope labeling triggered acquisition. And the way this works, if you can see at the bottom, is you digest your peptides and then you add heavy peptides, which are peptides of your protein of interest that are labeled with a very, very specific group, and you know exactly what that M/Z difference is. And what you do is you run that and you watch for it in MS1, and you tell the mass spec to watch for it, right? Because it's M/Z, its retention time should be exactly the same as the unlabeled peptides. And because you know the M/Z difference, you can look at that peak in the chromatography and just subtract the heavy peptide mass, and you can see your peptide. This was actually very promising because we were able to see not only the heavy peptides in our chromatography, we were able to see our peptides of interest in chromatography, which is huge because, again, we want to be able to run this on the mass spec so we can do turnover rate calculations. Now, the downside, and it's still an optimization thing, is that Peak's still couldn't recognize it. That's because we still got to optimize the mass spec. We've got to change some scan rates. We've still got to change, call it fill the bucket. We've got to change some key things in order to better. But the fact that we could see it in the chromatography and we could see it in our MS1 scans really suggests that we're getting very, very close on this one. But even after all of that effort, our outlook's not looking too good. And so what do you do when you can't see your target protein? Because we've got some appealing and some optimistic methods, but still nothing's worked reliably, or at least reliably enough. So I created this idea. Well, it's not me, it's this idea. I call it black box proteomics. It's this idea that if you cannot see your protein of interest, you can look at everything else you see, because we did good data. We had good data, right? And so even though we can't see sphingosine kinase itself, we should still be able to see something changing in everything else. Now, this only works when you really know the mechanism. And without going into extreme detail, this mechanism is very well established in the literature. So I created a cell type heat map. Now, these proteins, the colors represent, the colors of the different blues and teals and purple, that represents different areas of this mechanism. But I wanted to see what's going on, because in one cell type, our PROTAC would work, and then it wouldn't work, and then another one it would work, and then never work again. It was very, very convoluted, right? Again, we expected it should work the same across because they all have the same E3 ligase, right? Wrong. We saw drastic differences in cell types based off of how many proteins were detected, their relative abundance, and you can see here just the relative differences and how drastic they can be between cells for a single protein. And what this tells us is that these pathways are not the same. Should have seen this coming, but we saw key proteins attributed to apoptosis, cell proliferation, metabolism, we can detect them, but they're drastically different between cell types. And so that led us to kind of doubt whether sphingosine kinase itself is actually going to be this target that applies to many different cell types. Furthermore, when we did some studies of our PROTAC, which is the PF543, and SKI2, which is just that inhibitor, that warhead of ours, we found that the PROTAC actually works worse than the inhibitor. And so even though our data is good, even though we found some promising detection methods of in-gel digestion and SIL triggered acquisition, because our heat map analysis revealed such drastic differences in cell types, and because of the unpredictability and ineffectiveness of PROTAC-mediated sphingosine kinase degradation, we really think sphingosine kinase is not a good target. Now, what does that mean? It means now we get to find a new target. We get to make a new PROTAC. We can figure out what's going on, but we can also do some phosphoproteomics. We can do turnover rate analysis. We can really figure out what these pathways are doing. And that's where we'll be going moving forward. So in conclusion, I'd like to thank the J.C. Price Lab and the David Michaelis Lab, especially the members of the J.C. Price Lab and Michaelis Lab that are part of the PROTAC group. I'd like to thank BYU and the Fritz B. Burns Foundation, the NIH grant, and US HUPO for the opportunity to present. And with that, are there any questions?
Okay, great job. Thank you so much,
Thanks,
Noah.
Noah.
Very interesting. I think you did a great job of putting a really positive spin on what could have been a frustrating outcome for you. So do you get to have a say in what the next target is?
So for me, actually, I'm now working because, like you said in my intro, I balance between Alzheimer's and cancer, and so I'm back with Alzheimer's now. But moving forward, yeah. So I won't decide on the target, but it's not that I can't. It's just that I'm doing something else now. So.
Mm-hmm. So it sounds like you're dropping sphingosine kinase as your target, but I'm just curious, did you consider doing targeted proteomics to
Yeah.
find it instead of DIA?
Yep, and we absolutely did, and it just didn't work. And again, there's a lot of data behind this presentation, so I didn't talk about everything we did, but targeted was one of them. And even though we had the retention times, even though we had the M over Z's, it's just I'm very confident that it's within our signal to noise ratio. And so the mass spec just cannot identify it.
Can I ask what instrument you're using?
So for this, we had some trouble with our Lumos. And so we started on the Lumos. So it has the Orbitrap and the linear ion trap. And then we moved to the Eclipse because it's just the Lumos, just newer. It's a little different, but not much. The key here is that we needed an Orbitrap because to do DIA effectively and to do, you need something that can really hold the ions well. And then we use the linear ion trap to actually do the MS2 scans.
I'm going to mute myself. My fire alarm's going. Go ahead, Jordan.
Noah, are there any non-mass spec-based proteomic approaches that might be amenable for this type of problem in the future? If you're having a hard time detecting your protein of interest on the mass spec, are there orthogonal techniques that are being developed, maybe like Olink or Nautilus's new platform? Could any of those potentially be relevant for what you're doing?
Olink has some potential to be relevant. We'd have to adapt. We'd have to change so much of our lab's general workflow. We'd have to change the software for our turnover rate analysis. We'd have to change lots of things, and we'd also have to get an Olink instrument to actually do that process. But that one does look promising. Nautilus, I visited with them in Portland, and they seemed interesting to me. Some of the specific kind of nitty-gritty details of our analysis wouldn't quite work, but I think could be adaptable. So the answer is maybe.
Okay,
Fair.
great. Thanks so much to all of our speakers. I noticed we've gone a few minutes over time. I apologize. I was listening so intently, I was not looking at the clock. So thanks again to Ali, Coleman, Wei Jing, and Noah, and to my co-host Jordan, and to US HUPO for hosting this webinar and advertising it. And we hope to do more of these. If you have any suggestions for webinars you'd like to see us put together, please get in touch with US HUPO, or you can ping Jordan or I probably through Spaces, or I think you're on Blue Sky, right?
Yep.
Yeah. Okay, great. Thanks so much, everyone. Have a great day.